Okay, and this is material for your exam too. So we covered chapter 4 in class. The first part of chapter 5, sections 5.1 and 5.2 we covered in class. And so right here you have section 5.1, and let's just go through here very quickly. We talked about the nature of the phospholipid molecules, that it's a modified fat or oil. We've lost one fatty acid chain and replaced that with a mineral group, which gives the head portion of the molecules an electrical charge and makes the head portion of the molecules hydrophilic. The tail portion is still fat and oil-like. They are hydrophobic, repelled by water. And so, because of the nature of this material, it just naturally forms a double layer. And so, right here in this diagram, what it's showing is this material, the phospholipids, naturally forms a continuous flat layer because of both the amphipathic nature of the molecules and their cylindrical shape. And this is in contrast to what you see over there where it says detergent in water, because there you have just one fatty acid chain, which gives that molecule a cone shape. So let's just slide on over here, and what we had, we looked at this part right here, the idea that the components of this membrane are continually moving around, but these phospholipids move around laterally. What they do not do is they do not invert themselves, okay? Because if they would invert themselves, they would be able to invert those proteins in there. Now, the proteins that we have, we looked at this, and the proteins that are embedded in the membrane, like this, like these right here, this one over here, those are referred to as integral proteins. They are integrated into the membrane. Proteins such as this one right here, that are setting on the surface of the membrane, right here, right here, right up here, setting on the surface of the membrane, those are called peripheral proteins. Now, these integral proteins, like the phospholipids, they are amphipathic. They have part of the molecule that has electrical charges and is attracted to water, is hydrophilic, but the interior of this protein matches up to the nature of these fatty acid chains. So the interior portion of the protein is hydrophobic and repelled by water. And that is what holds this protein in place. If it tries to go sliding out of this membrane, the part that's repelled by water will be pushed back towards the interior of that phospholipid bilayer. The other thing to notice is these carbohydrate chains always extend to the outside of the cell, the outer layer of the plasma membrane. Also, so these proteins with a carbohydrate chain, those are referred to as glycoproteins. We can also have a carbohydrate chain attached to fats in here. It's called a glycolipid. And then cholesterol, right here, here. Cholesterol is going to be a part of this membrane, and cholesterol acts as a fluidity buffer, essentially kind of acting like antifreeze. So looked at this experiment where they fused mouse and human proteins, and with the mouse and human proteins, shortly after fusing these, the mouse and human proteins were intermingled with each other, showing that these proteins and the components of the membrane are not locked into place, but they are continually moving around. And so they became just intermingled at random, very short order. Okay, this was the idea that these proteins are oriented in a specific direction. So the characteristic of the outer portion of the protein is different from the inner portion of the protein. So the proteins have to be oriented in the correct direction across that membrane. We looked at this idea of manufacture of these glycoproteins that will fit into the plasma membrane, and getting these glycoproteins into the right orientation begins way back here in the manufacturing process, so that during this manufacturing process the sugar chain is oriented to the inside of the endoplasmic reticulum, then the inside of the vesicle, inside of the Golgi complex, once again to the inside of the vesicle. Then the vesicle fuses with the plasma membrane, which then places this so that the sugar chain extends to the outside. Okay? So basically right here in the Golgi complex, sugar chain's going to the inside of the organelle. Out on the surface of the cell, the sugar chain goes to the outside of the cell. So all this ties back into the placement of that. Okay, so let's go to this page right here, and we looked at the nature of these proteins that are found in the membrane, and we found that you've got proteins that are involved in anchoring, that they are attached to both the extracellular matrix on the outside of the cell as well as the cytoskeleton on the inside of the cell. Then right here we have proteins that are involved in passive transport. That is, materials that cannot freely go in between these phospholipids. They have to go through proteins, but still they are moving from an area of high concentration to an area of low concentration, and the cell is not using any energy to control this passage. Then we also looked at, go down here. Okay, so we looked right here at the idea that there are proteins involved in active transport, where the cell uses energy to move materials across the membrane. There are proteins that are involved in enzymatic reactions, that is, reactions that are sped up by the enzymes. And then we looked at the idea that you have signal transduction. With signal transduction, you have materials such as insulin that will attach to a protein from the outside of the cell. That'll set up a series of changes that changes the activities within the cell, such as allowing the cell to take up glucose from the outside. Some of these proteins in the membrane are associated with cell recognition, and the example they're using right here is the way that your immune system works by using proteins, here in this case antibodies, for cell recognition. And then down here at the bottom we have the idea that there are some places where you have cell-to-cell contact, and there has to be a way for those cells to attach to one another. And so you can have proteins in the membranes of these adjacent cells that will allow the cells to attach to one another. Okay, and then that gets us up to this section 5.3, which is where we cut off in class, and we need to go over that for material on the exam yet. Okay, thank you.